In this tutorial, we will cover real-time PCR, also known as quantitative PCR or qPCR, which is widely used for gene expression analysis, validation of RNA-seq data, and copy number studies. We will move step by step from the basic PCR principle to fluorescence detection, CT interpretation, primer design, reaction setup, and finally statistical analysis and visualization of relative expression data. By the end, you should understand both the experimental and analytical side of RT-PCR to build this properly. Let us start from the foundation. What is PCR and how it works? PCR stands for polymerase chain reaction. It is a technique that amplifies a specific DNA sequence from a very small starting amount into millions of copies. It requires template DNA, two primer, nucleotide, buffer, magnesium ions, and a thermostable DNA polymerase. The amplification happens through repeated temperature cycling. PCR is fundamental to cloning, diagnosis, sequencing, and expression analysis. Now that we understand standard PCR amplification, let's see what makes real-time PCR different and more informative. Real-time PCR builds on conventional PCR but adds a real-time fluorescence measurement. Instead of checking the product at the end on the gel, we monitor amplification during every cycle using fluorescent agent. This allows us to estimate how much starting template was present. It is especially useful when working with cDNA to measure gene expression level. To see how fluorescence reflects amplification, we should briefly revisit the three PCR temperature setup. First one is denaturation. During denaturation, the reaction is heated to around 95 degrees Celsius so that double-stranded DNA separate into single-stranded hydrogen bonds strands. Hydrogen bonds between bases breaks creates two templates for copying. This step happens at the beginning of the every cycle and must be completed for efficient amplification. Incomplete denaturation leads to poor yield. Once the strands are separated, the next step is to allow primer to bind. This is the annealing stage. During annealing, temperature is reduced so primer can bind to their complementary sequence. This temperature is primer dependent and must be optimized. Too high temperature will not bind and too low non-specific binding may occur. Primer specificity at this step determines assay accuracy. After primer are correctly bound, the polymers can extend them, which happens in the extension step. During extension, typically at 72 degrees Celsius, DNA polymers add nucleotide starting from the primer and synthesize a new strand. Each successful cycle ideally doubles the product amount, leading to exponential amplification. Reaction efficiency close to 100% in is ideal for RT-PCR quantification. In real-time primer, this growing DNA is detected using fluorescence. So let's look at how detection works. Uh, Real-time PCR instrument measure fluorescence emitted during amplification. This increases in fluorescence corresponding to the increases of DNA product. The machine records the signal at each cycle and plot it. The detection chemistry between how fluorescence is generated. There are two major detection chemistry commonly used. One is dye based and another is probe based. Let's compare them. SYBR Green binds to all double-stranded DNA and fluoresce. It is simple and economical, but can detect non-specific product. Probe-based system use sequence-specific probe that fluoresce only when the correct target is amplified, increasing specificity. Choice depends on budget, specificity needed, and assay type. No matter which chemistry is used, both generate fluorescence curve, and from this fluorescence curve, we determine the CT value. Now, RT-PCR workflow. The workflow includes nucleic acid extraction, purity check, reverse transcription of RNA studies for RNA studies, primer validation, reaction setup, plate organization, RT-PCR run, and post-run analysis. Error at early stage affect all downstream results. After running the reaction, the first data output we examine is the amplification plot of replication. The CT or cycle threshold is the cycle number at which fluorescence rises above the background threshold. It is inversely proportional to the starting template amount. A, difference of 1 CT roughly represents a two-fold difference in starting quantity of the quantity under ideal efficiency. CT value are the raw number used in expression calculation. When we plot fluorescence versus cycle number, we obtain the amplification curve. Let's interpret the next. The amplification curve shows baseline noise first, then the exponential rise, followed a plateau. Only the exponential phase is used for quantification because efficiency is stable there. The plateau phase is not used because reagent become limiting. To obtain reliable curve like this, we must follow a structured experimental workflow. Now is melt curve. Melt curve analysis gradually increases temperature after amplification and monitors fluorescence drop. A single sharp peak indicates a single specific product. Multiple peaks indicate non-specific amplification or primer dimer. This step is essential when using dye-based detection like SYBR Green. Specific product depends strongly on the primer quality. So next we focus on primer design principles. Primer should be specific to the gene, avoid secondary structure, have balanced GC content, and produce short amplicon for efficiency. Exon-exon junction spanning is preferred for expression analysis to avoid genomic DNA amplification. With validated primer, we then organize how samples are placed in the reaction plate. Now, a clear plate plan includes sample group, control, housekeeping gene, and replicates. Labeling and layout reduce error and simplify downstream analysis. Include NTC and non-RT controls where needed. After planning the layout, we prepare the reaction mixture carefully. The master mix contain polymerase, polymerase buffer, magnesium, DNTP, and fluorescence dye. We add primer, template, and water. But remember, we add the template separately and make the reaction mix separately. So while we are plating the samples, we put our samples into the PCR tube first and then the master mix. And we have to keep track of everything. Preparing a bulk master mix improves consistency across the wells. Mix gently to avoid bubbles. While setting up reaction, some practical thumb rules ensure reproducibility. 1. Prepare extra mix volume. Use filtered tips. Maintain consistent pipetting. Avoid freeze-thaw cycle and standardize template input. Always run technical replicates. Once CT value are obtained, we convert them into biological meaning using relative expression analysis. ΔΔCt normalized target gene CT to a reference gene CT and compare it with a control sample. This gives relative expression as fold change. It assures similar amplification efficiency between target and references. To compare multiple groups properly, we apply statistical testing, often using R. Also we can use Excel for this analysis. There is no hard and fast rule. We replicates data into log format, perform ANOVA, and run post hoc tests to identify significant difference between genotype or treatment. For this to work smoothly, the input data file must follow a correct structure. In data structure, each group should have replicate ΔΔCt value arranged clearly in columns, clear headers, and consistent formatting prevent analysis error. Once data is loaded, we convert ΔΔCt value into fold change values. Fold change is calculated as 2 to the power minus ΔΔCt, means the variation are computed per group. Log transformation often improve interpretability and plotting. These summarized values are then visualized for interpretation and publications. We generate bar plot with error bar and significant letter. Publication plot require high resolution and consistent formatting. Visualization help communicate biological differences clearly. If results appear abnormal, we then moves into troubleshooting mode. Issues such as primer dimer, inconsistency, or flat curve can rise from poor primer design, contamination, degraded templates, or incorrect setup. Systematic checking helps resolve them. Following standard based practice prevent most of this problem. Always validate primer, check efficiency, include control, run replicate, and document everything. Consistency across runs is essential for uh, comparative studies. Finally, here are the tools and resources used for this workflow. Script and visualization resources are provided for reproducible RT-PCR analysis. Learners are encouraged to reuse and adapt them for their own datasets. This completes the full journey from PCR principle to RT-PCR data interpretation. Thank you all.